I too want to thank you for showing up. Now, some of you here, particularly my students, I think, are wondering right now, what does he have to say about desire? And particularly, what is the link between desire and circadian rhythms? And I have to say that there is no obvious link between circadian rhythms and desire. However, if you work hard, you can find some links. And some of the work that we're doing may shed some light on this link, although I'm not going to talk on drugs and circadian clocks. Okay, one of the links, indeed, this is an anatomical link. We're working here on the region of the brain that's called the limbic forebrain that contains many important structures such as the bed nucleus of the striatum terminalis, the hippocampus, the striatum, the basolateral amygdala, the central nucleus of the amygdala. And these regions of the brain are important in regulating motivation, emotion, learning and memory, locomotion, reward, as well as drug seeking, withdrawal, and relapse. So here is one link between circadian rhythms and addiction or desire. We have been, we've been working on these regions, and in particular, we've been exploring the oscillations of clock gene expression in these regions, which mean that these regions have circadian clocks. But before, and you can see here that this cartoon here was shown to you by Jane Stewart at the beginning of her talk. The dopamine system really impinges on these different structures where we have identified circadian clocks. But before I go ahead to talk about those links, I'd like to say a few words about circadian rhythms in general. Those circadian rhythms are those oscillations, daily oscillations in a host of functions. Virtually every function that you can measure in an individual, human or animal, oscillates with a period of about 24 hours. So sleep wakefulness, these are things that you're consciously aware of, but you're not aware of cognitive fluctuations or fluctuations in affective tone, in emotional tone, motivation, metabolic fluctuations, autonomic, endocrine, gene expression, and neural activity. All these functions oscillate with a period of about 24 hours. Here are two examples of circadian rhythms that you're not aware of. Temperature rhythm goes up and down. So your body temperature, if you measure it continuously, you'll see that it goes down at night and it goes up during the day. Cortisol is a metabolic hormone, a stress hormone. Cortisol levels go up and they peak in the morning and they go down throughout the day. So here are two circadian rhythms with different peaks. One peaks at the end of the day, one peaks at the beginning of the day. These rhythms are not driven by any outside force. They're actually driven by circadian clocks. They are driven endogenously. And circadian clocks are found in, as I said, most forms of life possess circadian clocks, including plants and bacteria and so on and so forth throughout the phylogenetic scale up to humans. What are circadian clocks? Circadian clocks are really molecular oscillators that are made of several clock genes. These genes have been identified just recently in mammals and flies, and we know that the genes that consist the clock in flies, in rats, mouse, and humans are the same genes. The clockwork of this clock, endogenous circadian clock, is quite complex, but it is based on interlocking transcriptional and translational feedback loops. These feedback loops lead to the generation of circadian rhythms in clock gene expression and clock control genes. So there are several basic genes that are called clock genes, and they drive additional genes, all in oscillations which, with a period of about 24 hours. The clock itself is cell autonomous. It exists in each cell of the body, but only in certain tissues it would be expressed in the whole tissue and drives local circadian rhythms. What about the circadian system? As I said, there are many circadian clocks in the body. the most important one is the master circadian clock that's located in the brain. In humans, it's located in the suprachiasmatic nucleus. And then there are many subordinate clocks or oscillators, slave oscillators, that are present throughout the brain and the body. So you see you have clocks in the lung, adrenals, in the liver and heart, and these control tissue-specific rhythmic outputs. Now, the master circadian clock is not a very precise clock. The period of the cycle of this clock is not exactly 24 hours. It's slightly longer than 24 hours in humans, whereas in mice, it's slightly shorter than 24 hours. So for the clock to give an internal representation of the solar day, in order to be an accurate clock, it has to be entrained, and it is synchronized with the light-dark cycle, which is exactly 24 hours. So projections from the eyes to this clock communicate photic information to the clock, and they reset the clock on a daily basis. Now, the clock itself has to synchronize the subordinate clocks throughout the body, and that's what it actually does. It induces internal synchronization by sending all kinds of signals, time signals, to the rest of the body, to those subordinate clocks. So those signals could be hormonal or neural or even behavioral signals. And they coordinate the oscillations of all other clocks in the body. Now, what's special about those subordinate clocks and different from this master clock is that these subordinate clocks can also respond to behavioral state, to emotional state, to nutritional state, reproductive state, as well as to drugs. So these subordinate clocks not only receive circadian information from the master clock in order to really represent the solar day, but they are also sensitive to many perturbations and regulations by a variety of hormones and transmitters and stimuli. So these subordinate clocks then, because they can integrate circadian information from the master clock and information from behavioral state, can then integrate this information and use this information to control rhythmic outputs within specific tissues in the brain and in the periphery. Now, when you perturb the circadian system, when you disrupt circadian rhythms, these are some of the causes, by the way, that lead to disruption of circadian rhythms. And you may be familiar with this one, which induces jet lag. But these are all conditions that can disrupt circadian rhythms. And these are the consequences. These are the disruptions, the changes that occur as a result of disruption of circadian rhythms. So you have changes in mood, in motivation, cognition, vigilance, appetite, gastrointestinal, metabolic, and immune functions. And you can, those of you who travel across time zones, for example, if you go to Europe, you go into a period of jet lag, which really represents uncoupling between the master clock and those peripheral oscillators or slave oscillators. And this uncoupling, this internal desynchronization, leads to those changes in mood, motivation, cognition, etc. So it's very important for psychological and physical health to remain internally synchronized with the environment. The rat is our model, and the rat has a circadian system just like the human, and the master clock undergoes synchrony with the environment. There is internal synchrony between the master clock and slave oscillators. Clocks in the periphery are affected by behavioral state, and indeed, those clocks can integrate then circadian and state information. Now, we study locomotor activity in the rat because locomotor activity is really the hand of the master clock. We can tell by looking at locomotor activity what is the time of the internal clock of the rat. So we put the animals in running wheels here, and rats love to run in running wheels. Now, rats are nocturnal animals, so they run only at night and not during the day. What we do is we record the running activity of the rat, and we collect the information on a computer, and we plot a graph or a chart, an activity chart, that really tells us what time is for the animal. So you see here that this area here, right here, is the daytime, and this is the nighttime. Each line here represents a day, and those little black symbols here tell us when the animal is active. And when the animal is inactive, there is nothing. So we recognize three different situations for the rat under these conditions. When the animal is on a light-dark cycle, and by the way, these are the same charts that you see here, but they are double plotted. Each day is plotted twice, and consecutive days are plotted this way. So when the animal is on a light-dark cycle, the clock isn't trained to exactly 24 hours. And you can see that each day starts at exactly the same time. Once the lights are turned off, animals go into the wheel and start to run. Here, the animal is placed in constant conditions, no temporal cues in the environment, no light-dark cycle. And what you can see is that the animal goes into the wheel slightly later each day, which means that the period of the endogenous clock, the natural cycle, is slightly longer than 24 hours, because each day the rat goes into the wheel, very precise, but slightly later. So here, this rat, the period of the internal cycle is about 24 hours and 15 minutes. So the animal will go in 15 minutes later each day, very precise. Then if you keep the animal in constant light, which is another condition where there are no temporal cues in the environment, but unlike constant dark, constant light disrupts the circadian clock, and what you end up with is an arrhythmic animal. So being in constant light is very bad for the circadian clock, and it fails to regulate circadian rhythms. Okay. Now, the master circadian clock in mammals is called the suprachiasmatic nucleus, as I mentioned before. It's a really tiny, tiny structure located in the base of the brain, in the hypothalamus, right here, right above the optic chiasm. This structure here contains about 15,000 cells, and you can imagine how small it is because the brain contains billions of cells. So it is a very tiny area of the brain, and this is the master clock of the brain. This structure, this bilateral structure, controls all circadian rhythms. And here's an example of what happens when this structure is damaged. So this cartoon shows you that the circadian oscillators, clocks, cells, that drive circadian rhythms are located here in this region of the suprachiasmatic nucleus. And when you look at behavior, you see that the animal free runs here. Very organized locomotor activity rhythm. Now, what happens is when you lesion this, when you put down electrodes and you burn these regions, animals become totally arrhythmic. So clearly, this area of the brain is crucial for the maintenance of locomotor activity rhythms. Now, the cells of the suprachiasmatic nucleus are really special because they are the only ones who can be cells that can be put back into the animal. So what we do, or what people do, is they put fetal SCN grafts. They take fetuses, they take out the suprachiasmatic nucleus, they take out the cells, and they inject the cells right into this location, and this can then rescue or restores the locomotor activity rhythm, as you see right here. So this is really the main clock of the organism. Now, what we do in our lab is track gene expression. We look at the genes that make the clock, and we choose to look at the gene called period 2 or pair 2, which is part of this circadian feedback loop right here. It's a crucial gene for circadian rhythms because if you mutate that gene, circadian rhythms disappear. So what we do, we use all kinds of molecular techniques to follow the expression of genes and pair 2 in this region, and this is how it looks. We use immunohistochemistry. And what you see here, every dot here is a cell that expresses this particular protein and probably contains a circadian oscillator. Then we take slices from this suprachiasmatic nucleus from different animals at different times of day. And what you can see is that the expression level of this gene in the suprachiasmatic nucleus goes up, and then it goes down again. So there is a rhythm of expression of this period 2 gene and protein, which oscillates with a 24-hour cycle. It peaks at around the transition time from day to night, and it's very low in the morning at ZT1, just 1 hour after lights on. The limbic forebrain. Here are 3 structures of the limbic forebrain where we have identified circadian clock genes, the oval nucleus of the bed nucleus of the stria terminalis right here, the central nucleus of the amygdala, and the basolateral amygdala. We have studied other structures, but for today's talk, I'm just going to tell you about these regions. So each of these regions have been implicated in a host of important functions, including addiction and relapse, learning and memory, conditioning, etc. These are the levels of expression of pair 2 in these 3 regions at a particular time of day. I'm going to show you now the rhythms that they generate. So in the oval nucleus of the bed nucleus of the striatum terminalis, you see again an increase, a rise in gene expression that peaks around the transition from day to night and then going down again. Beautiful expression, very clear. In the central nucleus of the amygdala, again, you see an increase, a rise in expression, and then again a decline in expression. And you can imagine this pattern of expression and the previous one are pretty much the same. They're identical. And indeed, the central nucleus of the amygdala and the oval nucleus are homologous structures. They are made of the same types of cells. They contain the same neuropeptides, and they play similar roles in a host of functions. And then the basolateral amygdala. Here you see an opposite pattern of expression, with expression peaking in the morning and very low in the evening, as you see right here. And this is the basolateral amygdala. And remember, this is the central nucleus of the amygdala. It's quite remarkable. The 2 regions that are just next door to each other oscillate with a different pattern altogether. It's as if when it's evening here, it's morning here. Okay, now we've looked at many structures, and these are the patterns that have emerged. For the suprachiasmatic nucleus, the master circadian clock, and these 2 regions, the central nucleus of the amygdala and the oval nucleus, rhythms peak in the evening. Whereas in other regions, such as the hippocampus, the basolateral amygdala, and the striatum, rhythms peak in the morning. And if you superimpose that, you see that the brain contains multiple rhythms with different phases and different amplitudes. So that's quite intriguing. I have to say right on that we don't really know, with the exception of the master clock, what these rhythms are in charge of. We like to say at this point that they are important for housekeeping. This is something you say whenever you don't know what they really do. But they're probably important for the regulation of rhythmic expression of tissue-specific outputs. Let me give you a summary of what we've done so far. I see that I'm running out of time, but I'm going to take another 5 minutes. So we have established that the suprachiasmatic nucleus is essential for the expression of rhythms in the limbic forebrain. If you take out the suprachiasmatic nucleus, if you destroy it, or if you keep animals in constant light, those rhythms disappear. So they are really subordinate rhythms. They are slave oscillators to the master clock. But we have done quite a bit of work on the effects of behavioral state, of feeding, of stress, and some drug work, as well as the effects of a host of mediators, stress hormones, gonadal hormones, thyroid hormones, dopamine, which was mentioned earlier, corticotropin releasing factor, another stress peptide. All these manipulations have robust effect on circadian oscillations in the limbic forebrain, but they not all do the same thing. So feeding and stress and drugs have a more global effect, whereas those stress hormones and gonadal hormones have selective effects on some structures, but not on others. Now I'm going to give you one example of what we've done with feeding, with food deprivation that Uri talked about. So this is what happens when animal is given free access to food at all times. So they show normal circadian rhythms in the running wheel, and the suprachiasmatic nucleus is the clock that regulates these locomotor activity rhythms. Now what we do is we restrict food access to these animals. We put them on food deprivation, and we let them have food for 2 hours each day. And what happens is that these animals start to develop another rhythm of activity, which is called food anticipatory activity. And it starts at about 2 to 3 hours before the scheduled meal. And it starts very accurately without any external signal. This is another clock that is not located, by the way, in the suprachiasmatic nucleus. It's located somewhere else. We don't really know where it's located yet, but it's another clock that is entrained by the scheduled feeding, and it induces those food anticipatory running. It is also associated with anticipation in hormone release and body temperature. Body temperature goes up here. Hormones that are important for metabolism go up right before scheduled food. But this is an anticipatory rhythm. So these animals actually have two types of rhythms. One driven by the suprachiasmatic nucleus, the master clock, which is enslaved to the light-dark cycle. And the other rhythm is a rhythm of anticipatory behavior to food, which is controlled by an elusive food-entrained clock somewhere in the brain. Now, we looked at those schedules and tried to see what happens inside the brain in terms of gene expression. Oh, by the way, when you put animals back to normal feeding, food available at all time, they immediately go back to show only one pattern of activity and rest, which is driven by the master clock. Now, to look at gene expression, I'm going to superimpose that on this activity chart. So when the animals get free food, as I mentioned before, rhythms in the suprachiasmatic nucleus, the central nucleus, and the oval nucleus peak in the evening, whereas rhythms in the basolateral amygdala and the hippocampus peak in the morning. But when you put them on restricted feeding, everything changes. Although the suprachiasmatic nucleus remained enslaved to the light-dark cycle, all other rhythms become now entrained to the food, and they uncouple from the suprachiasmatic nucleus and establish a new phase which occurs about 12 hours after food presentation. So food is a very powerful modulator of clock gene expression in the limbic forebrain, in that region of the brain that is strongly associated with drugs. This procedure is so powerful that even if you take an animal that is totally arrhythmic because it has been kept in constant light, and you put the animal on this restricted feeding schedule, immediately you see that the animal becomes rhythmic again. It again shows locomotor activity rhythms. And not only that, but whereas under constant light, there are no rhythms in clock gene expression, when you put the animals on feeding, there is a rescue of all these rhythms, but this time even the suprachiasmatic nucleus become rhythmic. So feeding schedules, restricted feeding schedules, have very powerful effect on clock gene expression in the brain. One last experiment that I want to tell you about is the experiment where we looked at the effect of food deprivation versus daily access to a treat. So daily access to a treat means that these animals have free access to normal food, but once a day for 2 hours during the light phase of the cycle, they get access to a treat, which is chocolate and sugar. And what you see here is that both groups of animals show some anticipation. This one, of course, shows anticipation, as I showed you before, because this group is food deprived, get food only for 2 hours each day. They show this food anticipatory behavior, whereas these that get access to a treat, some show anticipation, some don't show anticipation. But overall, they eat about the same amount of food, by the way. Now we look at gene expression, and you can see here in these graphs that restricted feeding, induces this shift in clock gene expression, as you see right here, as I showed you before, and right here, and right here. But those animals that get access to a treat but are not food deprived are not very impressed with that, and gene expression in these brains do not shift, which means that in order to control gene expression by food, you have to be food deprived, or something about your homeostatic balance in the brain is the factor that affects gene expression in these regions. So I'm going to finish here, as you can tell. I want to acknowledge the contribution of my graduate students, most of which are right here, and my collaborators at Concordia, Dr. Jane Stewart and Barbara Woodside, and other collaborators from McGill and elsewhere, and of course my technician Barry Robinson, who's the man. He's the man. And I also want to thank Graham Carr for organizing this great symposium. Thank you very much.